Okay, so today we've got a great story for you. It's a story of scientific detective work, all about a brilliant technique that was developed more than 100 years ago to unmask one of humanity's oldest and deadliest microscopic enemies.
So let's set the scene. Imagine you're a doctor back in the 1880s. People are getting sick with this awful lung disease, but when you look at their samples under a microscope, you can't see the culprit. You know something is there, but it's totally invisible. So how in the world do you fight something you can't even find?
And this is the enemy, Mycobacterium tuberculosis, the bacteria that causes tuberculosis. Now, the only reason we can see these tiny, bright red rods so clearly against that blue background is because of a really ingenious staining method, one designed specifically to get around their natural invisibility cloak.
To really get why this solution is so brilliant, we first have to understand the problem. And the problem? Well, it lies with the bacteria itself. It is just incredibly stubborn.
See, these bacteria, like Mycobacterium and its relatives, they have this unique defense system. The best way to think about their cell wall is like a suit of waxy armor. And this armor doesn't just protect them from our immune system, it protects them from the very tools scientists were trying to use to see them. The secret ingredient in this armor is a stuff called mycolic acid. It's this really fatty, waxy material that makes the cell wall basically waterproof. But here's the crucial part. It also makes it impermeable to standard lab dyes, like the ones used in the gram stain. The dye just can't get in. It kind of bounces right off. And that is why these bacteria stayed invisible for so long. So, faced with this waxy fortress, how did scientists finally break through? Well, they developed a clever chemical assault that could breach the armor, and once it was inside, it would hold on for dear life. The whole process is this brilliant three-step trick. First, they use a super potent red dye called carbofuscin, and often they'll use heat to literally force that dye past the waxy mycolic acid layer. Second, they wash the slide with a strong acid-alcohol mix. Now this washes the red dye out of everything else, but not the stubborn bacteria. And then, for the third step, they add a blue counterstain, which colors all the other cells and all the background material. And this is exactly how the technique got its name. The bacteria that hold on to that red dye, even when they get washed with acid, are called acid-fast. The color is literally holding on fast against the acid wash. Pretty clever, right? And the result is this. Just an incredible visual payoff. The invisible enemy is suddenly completely unmasked. You get these bright red acid-fast bacteria that just pop, standing out so vividly against that blue background of other cells. The target is now in plain sight. Of course, science is never static. You know how it works. Once this breakthrough was made, researchers immediately started to tinker with it to refine it and improve it, and that led to a couple of key variations on the technique. It all started with Robert Koch back in 1882, but science is all about building on what came before. Soon after, two researchers, Ziehl and Neelsen, refined the process using heat to really drive that stain in. That became the classic hot method. Then, fast forward to 1915, a guy named Joseph Kinyoun figured out a way to skip the heat altogether, just by using a more concentrated dye. And that gave us the cold method. Each step just made the whole process a little easier, a little more accessible. Okay, so you might be thinking, a stain from the 1880s? In the age of DNA sequencing, that's got to be ancient history. So where does a classic technique like this fit into the world of 21st century diagnostics? Well, modern diagnostics has seen this huge shift. We've moved from what we call phenotype methods, identifying microbes by what they look like or how they act, to genotype methods, where we identify them by reading their unique genetic code directly. It's kind of the difference between identifying a person by their photo versus using their DNA fingerprint. And the advantages are really clear. Molecular methods like PCR are incredibly fast. They give you results in hours instead of days. They're also way more sensitive. They can detect just a few bacterial cells in a sample, whereas staining and culturing need a whole lot more to get a positive result. And yeah, because of this speed and sensitivity, these nucleic acid tests like PCR are fast becoming the new gold standard in a lot of labs. They're either replacing or, at the very least, working right alongside these older classic techniques for diagnosing infectious diseases. So, if we have all these powerful genetic tools, why are we even talking about a 140-year-old staining method? Does it still have a place? The answer is a resounding yes, absolutely. And the reason why it still matters so much comes down to this number, 1. What could possibly be so important about the number 1? Here it is. The power of this technique is so profound that finding just a single red acid-fast bacillus in a patient's sample can be enough for a diagnosis of tuberculosis. One single cell. That is an incredible, almost unbelievable level of diagnostic power.
And that's why its legacy is so strong. It's incredibly reliable. It doesn't require expensive, high-tech equipment, just a standard microscope that you can find pretty much anywhere. And this makes it absolutely essential for diagnosing TB in resource-limited parts of the world. It provides a fast, cheap, and effective first line of defense against a major global killer.
You know, this whole thing points to a bigger idea. While we're all chasing faster, more complex technologies, the acid-fast stain is this powerful reminder that sometimes the most elegant and enduring solutions are the ones that are simple, robust, and accessible to everyone. It really makes you wonder, what other simple tools are out there quietly changing the world every single day?